This is the last discussion for today, and it will wrap it up our discussion about biotech and space. And we have as guests for this Elliot, Roth, and JJ. And I would like you to, I don't know who I can introduce first. I think I'll start with Elliot, because I see that Elliot is very excited to start. Would you like to say some words about your work,
Sure.
Elliot? And
I'd also like to say that another member of our crew at High Seas is here too on the call. So Jessica Snyder was also a member of our crew. I don't know if she's on video. Yeah, there she is. Hi. So the three of us are going to be...
Welcome.
Can you hear me all right? I'm sorry, I literally just got off my flight in Dallas. So if you can hear me, fantastic. And now I'll step back. Thanks
so just a brief introduction. I have a background in algae technology and whatnot, and I was lucky enough to be brought into this realm of analog space missions by JJ. And JJ, take it away with an introduction.
Sorry, I have to take off my mask because I'm talking in the airport. Let me know if you can't hear me at all. Yeah, so I'm JJ Hastings, now doctor. 4 years later, since you guys knew me, I'm now a doctor. Woo! Yeah, so I have a company now. After starting my very first analog mission and getting into space simulation as a practice back in 2018, I now have a company, Analogica, that is leveraging the simulated space area as a way to do research. But I also have spoken, thank you Nina, shout out to her who's on the call, spoken on hacking for space and how you can use the resource constrained environment of the analog space itself, the habitat, to kind of drive innovation. So that's where I'll step back.
And Jess, you want to introduce yourself too?
Sure. So I'm a mechanical engineer. I got a PhD in biofabrication about 5 years ago, and since then I've been working on biological industrial design. And I think I had more fun than you're supposed to have on a 2-week analog mission, and I credit Elliot and JJ for all of that.
So already we have a kind of question about analog missions, and I'll do my best to kind of answer that. I have a brief presentation to share that I think, let's see, if you can see my screen. Can everybody see my screen now?
Yes, we
okay.
can see it.
All right, cool. So an analog mission is basically a simulated space environment. What that really means is that Jess, JJ, and I lived in our home sweet dome, this domed habitat on the side of a volcano in Hawaii. It is a teeny tiny little space. As you can see, this is kind of like proof that I was actually there, standing on the side of a mountain. This is the floor plan of the dome itself. I think, and correct me if I'm wrong, JJ, it's like 35 feet across, or about 10 meters, a little over 10 meters across.
Yeah, that's right. It's 38 feet tall is the radius. So if you take 38 feet right above you and then extend it out all around you, that's the radius of the half dome.
And it looks like a golf ball almost cut in half, and it has like a little second level. And the purpose of analog missions are really to test the it tests different technologies in a space-like environment without actually having to go into space. So our little dome is called High Seas, and it's part of the International Moon Base Alliance. And they do a lot of research in conjunction with a variety of different space agencies, but mostly to test technologies that would enable life on other planets and really test the squishy bits that come into play when you have a group of people, very high-performing people in a tiny space for a prolonged period of time. So let's see. This is what it looks like on the inside. It kind of has this like puffy, billowy, cloud-like interior. Each of us had desks in a little rotunda around the edge. And it's kind of like living in a life support system. So this habitat, you enter into these habitats and you have to stay there without leaving for certain periods of time. And when you go outside, you have to wear a spacesuit. And so I'll show you kind of what that means. These are the spacesuits. I'm showing a picture, JJ, if you can't see, of Paul and Michaela outside the habitat kind of waving. So anytime we went outside, we had to wear a spacesuit. And we were tasked by NASA Goddard, particularly with this mission, to investigate and explore lava tubes as a means of finding ways that people could live on other planets. And so the assumption is, especially on the moon and Mars, is that we'll have to live underground to help with radiation shielding, and that people will live within these underground tunnels. And so we actually went into these tunnels. JJ can talk a little bit about some of the other sort of things that she ended up doing in these tunnels. But this is a picture of our mission commander, Michaela Musilova, taking different bacterial samples of extremophiles that we were trying to find and identify in the tunnels. JJ, you want to talk about some of the other stuff that we did while exploring?
Yeah, sure. So one of my favorite aspects about high seas, so I've been in a few different analog spaces, and there are a few that are operated by different space agencies, like NEK outside of Moscow, that's operated by Roscosmos, and Hira, which is operated by NASA, well, by Wiley Laboratories, but officially NASA, out of Houston. So what I love about high seas is that you have these extraordinary networks of lava tubes that when you get into your EVA suit, really give you an otherworldly feel. The EVA suits definitely give you the impression of being completely constrained, and the air is moving. You are sensitive to just how much energy you're expending. Elliot can tell you about lots of fogging up of the visor, poor thing. But in the lava tubes, we do a bunch of different experiments, including doing air quality sensing, LIDAR, so spatial measurement of new cave formations, so new to us, and of course, different microbial studies as well.
Yeah, we also had a chance to test out one of the BioSummit community's product lines. So Nyoka Page from Nyoka Labs, she has a bioluminescent glow stick. So we tested that, the luciferase enzyme underground in the lava caves. And this is me kind of like testing out different stuff. As you can see, we are kitted out head to toe. Every single part of our bodies are covered. So motorcycle gloves, a helmet, flashlights, and we are normally kitted out with GoPros or cameras of some sort. We had an air pack and air tubes, and you can kind of see the condensation and fogging on top. And that's what happens if you are exerting yourself quite a bit. So this is another picture of the bioluminescent light. And so my background in algae was as a means of growing food in these sort of space-like environments. And so I tested a number of different growth experiments growing algae on wastewater and urine and other things like that, simulated urine. Then this is actively our laboratory. We had a little lab here. And Jess, do you want to talk a little bit about some of your experimentation while we were here and kind of what we collaborated on and stuff?
Sure, thanks, Elliot. My background is all tissue engineering, using 3D printing to make organs and tissues in a lab environment for people. And while doing that work, I thought there's a lot of opportunity here to use the same kinds of technology, the ability to design computationally, the ability to prototype using living cells, to do that for a whole range of things aside from just the strict rigors of what you would need medically. So in working with Elliot, we thought about how we could use agricultural waste and mycelium spores and 3D printed forms to try to grow as much as build useful products. And so it was Elliot's good idea to start with material testing. And so we created tensile molds and also compressive molds that we loaded or didn't load, so a couple parameters to explore there. The other thing we thought about was creating a hardware store. Can we grow a hardware store in our simulated lunar habitat? And so using 3D printing again, and a 3D printing filament that's a blend of wood particles and PLA, created scaffolds, seeded them with agricultural waste and with the spores to see if they grew to become load-bearing functional elements, and that's still growing, and so we'll see the results at some point. But those are the kinds of ideas that we were thinking about, how to use a combination of the locally available material, the resiliency of biology, and then some guiding factor, whether that's a 3D printing form or compressive force or something like that. Those are the kinds of things that Elliot and I were working on while we were in the habitat.
And some of the other things that we tested and kind of investigated were biometric style data. And so this is a number of biometric tests ranging from a TENS unit, which is an electrical stimulus, then a couple of different blood glucose measurements, some sleep tracking, and of course, variety of different cooking experiments. And so JJ was really one of the queens in the kitchen, did a phenomenal job cooking, as well as testing a number of really interesting factors when it came to biometric data. So JJ, you want to talk a little bit about biometrics and different sorts of microbial tests and other things that you ended up doing, as well as some of the more fun dishes that you made?
Yeah, sure. What's so funny? So this mission in particular was lovely for me because I'm used to being the one that's organizing and commanding and having to worry and fuss about absolutely everyone else. So this mission was fantastic in that I could enjoy just cooking and spending some time in the kitchen and just going out on EVA and not having to worry about everyone else. So I'll say I got to do a lot more cooking, which is so much fun. It's challenging in this environment, but it is so much fun. So doing kitchen bio is just, I highly recommend trying to work through different experiments for long duration space missions. So exciting. The other thing to consider here is what I call the sort of invisible elephant in the habitat, and that's microbial life. Sorry, I'll just lift my mask here a little bit so you can hear me a little bit better. So the microbial life that's in that habitat and the selective pressure that's placed upon it once we are sealed into that environment. And since we can go out on the crater, out on the lava field, what we might bring back and might contribute to our internal symbiosis or dysbiosis of microflora. So that's the longer term plan for my company and what I do through some of these missions, which is do surface sampling, so environmental sampling, plus doing sequencing or omics on a whole bunch of other biotic life in the habitat, plus a whole bunch of other biomarkers, but that's for the more sophisticated missions that we run with NASA.
Yeah, so the idea is to kind of like track people over time as they experience life in the habitat. You're in this enclosed environment. You're definitely interacting with people. All of us, we only had 30 seconds of water, of shower time every single day. And you have a limited amount of water. You have a limited amount of power. The CO2 levels are high within the habitat. And you can only eat freeze-dried food while you're there. And then in addition, we have to exercise at least 30 minutes a day. And we have 2 windows looking outside, and that's about it. So you have to exercise if you're not going on EVA. And there was some really kind of rough food experiments. This is a picture of me with the egg replacement. It didn't turn out so well. And so we learned how to work with freeze-dried food quite a bit. And I think even some of the crew members just didn't end up showering, just found other ways of really cleaning yourself and hygiene and other things like that within the hab.
Baby wipes. I didn't take a shower the entire time, plus a few other days on either end, and baby wipes are actually amazing. I recommend trying it at home or out camping. Highly recommend.
Yeah, it was actually surprisingly easy to not shower once you get into that mindset and you realize how much you actively consume. We did some more like esoteric experiments as well. So there did some fun things with slime mold that Jess is probably going to talk about in a little bit. So this is a picture of the slime mold style experiments we were doing kind of with the island of Hawaii. And some of the building materials, building different things on the moon using simulated lunar regolith, as well as compressive and tensile tests. This is a water purification and reclamation device. A donut drone that was tested. Basically, the sky's the limit. Anything that you can think of in a space-based context is what we would want to test in this sort of environment. So big thanks to the entire crew and everything. And I'm going to pass it along to Jess to bring us home.
Oh man, no pressure. So thanks, Elliot. I guess this is a picture that I like to sort of think about and look at. And although this is not a live picture of high seas, I think in looking at it, we can get a sense for what life on the moon might be like in maybe a couple decades or so. And I think like key things to notice is the existence and the presence of living things, familiar living things. You can see trees and plants, people taking care of those trees and plants. And that's the hope, that's the future that I would hope for, is that we can bring a lot of the things that provide us nourishment and familiarity from Earth and bring them to the moon. One thing I want to point out is there are two what looks like closed circuit video cameras in the kind of the upper left-hand corner. I think you can see those. And while automation and technology and observation and objective situational awareness, reliability, all those words are important, especially if we're going to be doing something like setting up a lunar habitat. But let's remember that we want to balance those kinds of invasive measuring with also the human perspective and people's need for privacy and their ability to make good decisions in the moment too. So if we just advance to the next slide. So like this happened in early 2019, late 2018, and it was the Chang'e 4 probe that landed on the far side of the moon. It was a Chinese mission from the Chinese space program. And what the mission did is scoop up a small amount of regolith, the soil on the moon, and then seed a couple types of plants. And the cottonseeds germinated. They became plants. And this was the first example of getting closer to that lunar habitat we saw in that past picture. So we're on our way. The first shovel's been shoveled. So if we go to the next slide. So in the spirit of trying to grow, integrate grown and born and made and controlled and all those different parameters, biology and technology, one thing I looked at was trying to use scaffolds to grow or guide the growth of microgreens. And if there's something about using a controlled process to create a planter that would help you produce something in a way that's more efficient, either from a soil perspective or water perspective, and kind of an open question. And growing things is kind of fun. So I tried this in the habitat itself. And so the results that you're seeing on the screen were done in my own apartment in San Francisco. In the habitat, I found it was much too dry, and the temperatures oscillated from very cold at night to rather warm during the day. And so because of that, keeping the plants from drying out was an obstacle, and the growth never really took off the way you can see it here. So it's just something to know about the future, the importance of irrigation and control when we move to these more closed systems, I think will just become a bigger factor. And also being able to use 3D printing to create ways of organizing them and maybe irrigating them, those kinds of things. This is a picture from some of the work that Elliot and I were working on. It's the agricultural waste seeded with the spores mixed with some of the lunar regolith simulant to see if we get different mechanical properties by varying the ratio of agricultural waste to that lunar regolith, and what do the spores do in response to that lunar regolith. So next slide, maybe. And then also kind of playing with 3D printing. Again, we can make these parts, but I'm really interested in sort of thinking about 3D printing assemblies, or even 3D printing interconnected linkages, getting closer and closer to maybe textiles or hanging elements. And if we could imagine a future where all this stuff kind of comes together, where you could create these woven elements that maybe hang as curtains or privacy screens, and then seed them with these microgreens that gives you some familiarity of Earth, you could start to think about how that might really improve life support systems or just your experience living in a habitat for a prolonged period of time. And then this is the slime computer. In talking to Elliot and JJ and thinking about this even before the mission, you know, the differences between what is alive and what isn't, kind of a fun question to think about and talk about too. But if we think about like the current state of the art and origin of life research, I guess what people kind of are saying is that first there was physics, there was the Earth, it was a big rock. And then over time, that big rock, there was big physical events, bombardments, things like that nature. And that created an avalanche of increasing chemical complexity that eventually resulted in biology and life. So it means that biology is an emergent system from both physics and chemistry. So if we start to think about biology as being somehow unique or different than what decisions would happen biologically or chemically, then can we compare the flow pattern or the growth pattern from the slime from the top of the volcanoes towards their food oatmeal? Can we compare that to, say, how the lava would flow or how water would flow? And it's just kind of an interesting experiment. So what we did is Elliot found a scale model, 1-to-1 scale model of the big island of Hawaii on Thingiverse. We printed it out with this wood polymer PLA blend 3D printing filament, and then seeded the slime molds in the tops of the two volcanoes on the island. We put the attractor, the food, around the outside of the 3D printed model. We saw some growth, but we didn't see very much. I think it was the slime didn't necessarily want to grow in that material. So inconclusive results, but maybe something we'll visit in the future, is how biological pathfinding compared to, say, geological pathfinding.
Yeah, and I would have to say, like, being able to use something like microbial, something kind of really, really small as a means of finding that pathfinding, or being able to find resources in these low resources settings. It's really intriguing possibilities. JJ, you want to bring us home with a call to action of how people can get involved in these kind of simulated analog missions?
Oh, fuck yeah. So I think, as you can see, part of the advantage of going into these analogs is that you're forced into completely new constraints and new environments. And part of that is getting the right crew in there, right? Jess and Elliot were phenomenal in that they came in with so many different ideas and then just got at it and kept working at it. And that's the essence here of hacking for us, right? So in the future, if you're interested in getting involved in analogs, I'm happy to just introduce you to the people. So if you want to organize your own mission, you can. If you have experiments that you want to take in on the mission, I'm happy to talk to you about that. Elliot can talk to you about his experience, kind of bringing in other people's experiments to work on. Jess can, oh my gosh, Jess was amazing at documentation and thinking about how to problem solve just over and over and over again. So Jess would be invaluable for advice on how to succeed in really tough conditions. And the other thing too is just to self-promote. I have this new Sensoria program, which is meant to elevate the position of women and persons of color, other historically disadvantaged groups in the space sector. So we provide professional development, opportunities to go into the habitat together, all that fun stuff. So please join us for that.
Perfect. And I think we have some time for some questions as well. So I'm just going to read through some of the questions. We got 5 minutes for about questions. So did you try cultivating microalgae to reduce CO2 levels, produce more oxygen? Yes, that was part of the point. Was we had a CO2 sensor that either was broken or the pump wasn't working altogether too much. And so we tracked average CO2 levels over time, and it seemed like the CO2 levels on average dropped from about 1,000 ppm average at the beginning to about 700 at the end of the mission. And so I don't know whether or not we were just not breathing as hard, or if it was the algae actually doing something, but I like to think that the algae helped.
So I had a question for Jessica, especially about bioprinting. So I'm avid in 3D printing as a hobby, but I'm also slowly getting into bioprinting. Can you comment more about like some really useful bioprinted objects that you've made on your mission? Like, what do you think has been like the most, like your favorite, I guess, and easiest thing that has actually like worked?
You are asking the $1 million question. I think it's one of the cases of, I think sometimes with technology, you can, if I want to scratch my left ear, I go like this, right? Well, sometimes with 3D printing, I feel like we're trying to scratch it like that. We're trying to use a technology because it's fun to use, not necessarily because it's the best. So that's why, to answer your question, I think that my favorite thing that I made was the interlocking flexible thing that you saw, because if you can start 3D printing these textiles, they not only don't take up like a horizontal space, we can actually hang them, so it's a little bit easier to incorporate them into the habitat. And these assemblies that sort of function on their own, and you can incorporate biology into them, I think are the most interesting. I've been working on sort of making these like little planters full of chia seeds, and can you do that with other plants, like maybe orchids or vegetables or whatever that is, moving in that direction, integrating 3D printing with growing stuff afterward. I think it's the most, there's a lot of room there, I think, to explore. And that's not necessarily bioprinting, but maybe it's sort of coming at it from a slightly different angle, but ultimately similar results. So I don't know if I answered your question, but I think it's a good one. And if you come up with good answers, I'm excited to see what you make.
There's another question from Nicholas about if we would do it again. And for me, that's a yes. What about the two of you?
Yeah, I mean, I'm going back in December. I was supposed to be there for a month and a half, but I'm off to Taipei instead. So different kind of isolation and quarantine in a hotel room instead of with my amazing crewmates. Yeah, absolutely, 100%. Highly recommend. Someone asked about the right characteristics. Personally, I would say patience and just simply openness. Understand that, you know, understand and be humble about what you don't know, and that things aren't going to happen exactly, including your own body, because you're at altitude and you can find space and everything's new, different diet. There's so many new variables for you. So coming in with humility and patience and just openness, I think are really advantageous.
I do have a bit of a question here. Given your experience, what did you find to be the biggest constraints to engineering in the environment?
You kind of have to learn how to hack it, to be honest. So like one of the big constraints is the availability of resources. And so the least amount of resources possible enable you to really get creative with what resources you have. So if you don't have something on hand, like for example, I didn't have all of the chemicals necessary to create a like accurate urine simulant. And so instead, what I did was I analyzed like what was in the yeast extract that I had on hand, and then used that as a substitute for some of the chemicals. And so you sort of have to make approximations. Oftentimes, it's not verifiable data or other things like that. Like there's hard decisions that you have to make as a means of approximating the answers that you can then go back and do. And that kind of is why these sort of analog missions are really valuable, because it gives you a test bed without the risk of like, oh, I went into space, but I forgot something, right? So you're able to really like roll with the punches. I would say in the future, I would like to do more DNA-based experiments, more synthetic biology in that kind of environment. It's a little bit of a challenge to do stuff like that without a hood on hand. And so that was the big challenge.
Thank you.
I don't have a question, but I do have a comment. In general,
I'm actually going to interrupt you guys and say that we need to shuttle you guys to the ending session, and you guys are going to have to take comments into Slack. So I'm going to take meta lead approval. This is Maria, and say this is great, but we have started the ending session, so somebody can put the link in the Slack. Please, keep asking JJ, Elliot, etc. questions. This has been a fantastic session. I was so excited about it. The analog space missions are amazing. But please come join us for the ending session, which is in Nucleus and has just started.
Thank you, everybody. And if you do want to connect, go to sensoria.space to check out upcoming analog missions. Repping you, JJ. And then, yeah, please connect
Thanks,
with
Elliot.
me, Elliot at Spira Inc. I just dropped my email in the chat, and then I'll also be on the Slack, so feel free to message.